Hi, everyone. Welcome to another Buena Vista Museum of Natural History and Science Meet the Expert event. I'm Mark Hodson with the museum, and today we're here with Dr. Anthony Rathburn. He will be speaking to us about his trips to the seafloor in a submersible vessel. Dr. Rathburn is currently professor and chair of the Department of Geological Sciences at the California State University, Bakersfield. Also research associate with the Scripps Institution of Oceanography, the interim director of the California Energy Research Center, and the director of the California Well Sample Repository. He comes to these positions with a bachelor's degree in zoology and a master's degree in geology from the University of Vermont, and a PhD in geology from Duke University, with additional experience as a postdoctoral research scientist at the Australian National University and also at the Scripps Institution of Oceanography. He was interested in fossils and started as a technician working with a marine micropaleontologist who studied both living and fossil foraminifera. Foraminifera are a single-celled organism that make shell-like skeletons called tests that commonly are preserved in the fossil record. He has been hooked on studying these mysterious tiny creatures ever since. Foram fossils hold clues to the ancient environments they lived in, and he enjoys the detective work of figuring out these clues. Foraminifera live in every corner of the world's oceans, and his work has taken him all over the world to study them. His affinity for water appears to extend to the frozen variety, as he has lived and worked in Antarctica for a year, first as a general assistant and then as a power plant operator. More generally, he enjoys traveling to remote areas of the globe, and since seawater covers most of the Earth's surface, his chosen specialty of oceanography is a good fit. Before we get started, if you enjoy today's event and you would like to see more content like this from the Buena Vista Museum, I encourage you to make a donation. You can donate to the museum anytime at buenavistamuseum.org/donate. I'll put that URL in the chat box for you. Again, that's buenavistamuseum.org/donate. Please keep your devices muted, and if you have any questions during the presentation, please type them into the chat window. There should be a chat button on your screen. Just click that and enter your questions. We will address them at the end of the presentation. If you are unable to enter your questions in the chat window, you are welcome to stay with us until we've answered all the chat questions. At that time, I will let you unmute yourself and we will entertain your question. Without further ado, welcome, Dr. Rathburn.
Thanks, Mark. Welcome, everybody. Today, I want to take you on a couple of trips out to the deep ocean, in fact, to the bottom of the ocean, and introduce you to some of the creatures and some of the habitats that I've encountered there. Before we get started, I want to give you a little bit of a preview of what we're going to talk about. So first of all, I'll introduce the ocean and some of its properties, discuss a little bit about how we study the deep ocean and some of the sampling devices, including marine submersibles that we use to study the deep ocean. And then talk a little bit about what it's like to travel to the bottom of the ocean, including preparation for the dive and the actual dive itself. And then introduce you to some of those seafloor creatures and habitats that occur and that are prevalent on the deep sea floor. Then just briefly, I'll tell you a little bit about marine science and our marine science program at CSUB, and then hopefully answer any questions that you might have. So let's talk about the ocean first. Seawater covers about 70% of the Earth's surface, and the deep sea is the largest habitat on Earth. And it's larger than all of the jungles, all of the deserts, all of the prairies, all of the forests, all combined together. It's bigger than that, and yet we don't know a lot about it. In fact, we have better maps of the moon and now Mars than we do for most of the deep ocean. If we remove the seawater, which would be a bad thing, but if we do, you can see from this map that there's a lot of topography there. So there's mountain chains, there's valleys, and the water itself also is very dynamic. There's currents going every which way at all depths, from the surface all down into the deepest parts of the ocean. So it's a very dynamic place. Now, if we look at if we compare some of the features of the seafloor with some of the features on land that we're more familiar with, we can see that the ocean compares pretty well. So if we look at the Mariana Trench, which is the deepest part of the ocean, 11,000 meters, You can see how it compares with the height of Mount Everest. And I'm going to pick on Mount Everest again, because a lot of people think that Mount Everest is the tallest mountain on Earth, and it's not. If you measure from the base to the top of the mountain, then in ignoring sea level, then the tallest mountain is actually on the big island of Hawaii. In fact, that mountain from base to top is almost a mile taller than Mount Everest. Also, if we just compare some more local features, if we compare our Monterey Bay Canyon just off the coast here with the Grand Canyon, you can see in the upper left-hand corner there, the picture showing that it compares pretty favorably, that they're about the same size. And yet the Monterey Bay Canyon is a fantastic canyon, but it's not unusual, it's not uncommon, and it's certainly not anywhere near the biggest canyons that occur in the deep ocean. And so if we were able, again, to remove all of the water and we're able to explore some of these other canyons, we might rename the Grand Canyon as the not so Grand Canyon. Now, I don't mean to pick on the Grand Canyon. It's a beautiful place and everybody should visit it. It's an amazing place. But again, there's lots and lots of canyons on this planet. And so just to give you a few examples, if you look at the upper right-hand corner there, in that diagram that map is essentially showing the seafloor features off the coast of California. Actually, one of my former students was part of a team that created this map. That's part of his job now, actually, to go around the world and map in 3 dimensions seafloor features that include faults that create earthquakes, as well as submarine landslides and other features. The lower 2 diagrams in the slide here is showing other areas of the world where there's lots of canyons and many much bigger than the Grand Canyon, including off the southern coast of Australia, off Kangaroo Island, and on the Alaskan margin. These cruises, these research expeditions were ones where we were actually targeting seafloor samples, but also we wanted to map the seafloor. These areas had not been mapped in any detail prior to that. Now, one of the questions you might have is how do you do that? We can't really just take pictures like we could with a drone over a terrestrial environment. Well, because it's covered with seawater, we can't use the light because there's no light at the bottom of the deep sea, but we can use sound. So we use sonar basically, and we bounce sound waves off the bottom and we bring them back and they bounce back to the ship, and we can interpret them and figure out what the topography is of the seafloor, as you can see in the diagram there in the lower left. Okay, so let's talk about some of the properties of ocean water and of the oceans. Now, the maximum depth for a recreational diver is 30 to 40 meters, 100 to 130 feet. Now, you can, with special gear, you can dive a bit deeper than that, but the average depth of the ocean is 3,800 meters, or a little over 2 miles. So that's just the average depth, and that's way, way, way below any single diver's ability to dive. So we have to use other instruments in order to be able to study the deep ocean. We can't just use scuba gear. So what happens when we go down into the deep ocean? Well, first of all, light decreases as you descend. So all of the light is gone at about 1,000 meters. And between 200 meters and 1,000 meters, it's twilight. So it's getting pretty dark. So well above the average, the ocean below 1,000 meters is as pitch black, as dark can be. And of course, the water gets colder as well. The average temperature of the bottom of the ocean is around 2 to 4 degrees C. So around 35, 38 degrees Fahrenheit. So pretty chilly. And of course, the pressure increases because we have all of that weight of that water above us. So at the surface, we're exposed to 1 atmosphere of pressure. And then 10 meters down, or 33 feet down, we're at 2 atmospheres. And then at 66, we're at 3, and 99, we're at 4, and so forth. So you can imagine the amount of pressure that anything at 4,000 meters is exposed to. So anything that we put down to the bottom of the ocean has to be designed to withstand the cold temperatures, the lack of light, so we can't use a solar panel, and the pressure. And that's something that astronauts don't have to worry about. They don't have to worry about pressure on their capsule, but certainly we do when we're designing any sort of equipment that we're going to send to the bottom of the ocean. So what happens to something that is not designed to be at the bottom of the ocean? What happens to that when it's exposed to pressure? Well, if you look at me rather than the slide, here's a styrofoam cup, which is mostly air. So there's a lot of air pockets in there. And this is what happens to it when we send it to the bottom of the ocean. So this cup was this size before we decorated it and sent it to the bottom of the ocean off Alaska. Now, also, oceanographers, like all of the early explorers on terrestrial environments that were going to remote places, also have their collection of shrunken heads. So this was this size before we decorated it and sent it to the bottom of the ocean off Costa Rica. Again, the pressure squeezing all of the air out of the styrofoam. And here's a picture of some of the other decorated cups and decorated heads that some of our creative students created in between samplings during cruises. Okay, so what do you do when you're preparing for a cruise? What does it take? Well, the first thing you have to do is figure out, you have to get funding, for example, in order to fund this whole thing, which is not easy at all. But then you have to decide where you're going to go and what samples you're going to take and how you're going to take them and what gear you're going to need. Then you pack the gear, and as you can see up here in the top center, you get all of that gear and you load it onto the ship. And the research ships have empty labs, and so we have to set up our labs. And included in the setup, of course, is the idea that we have to tie everything down, like this microscope there in the center. As you can see, it's all tied down because we want to make sure that it doesn't fall off when the ship is rocking and rolling when we're out to sea. So everything has to be tied down and secured. We also have to get our sampling gear ready and set up. So we do that on deck, and get also our vials and our sampling bags, etc., all pre-labeled as best we can and prepared to collect samples. We also have to learn how to use some of the equipment on board, and so technicians show us how to do that. And then also we have to go through safety training, and one of the parts of the safety training is to make sure that your immersion suit is the right size. And of course, also we have to get familiar with the ship to know where things are, and we have to learn the ropes, literally. So what do we use to study the deep sea? Well, of course, the first piece of equipment that we really need is a ship. And depending on how long we're going to be out and how far offshore we're going to be dictates the size of the ship. The Sally Ride that's shown here is the newest ship on the Scripps Institute for Oceanography fleet. And this is a ship that we sailed on a few times now. And it can hold, as I recall, about 24 scientists along with the crew. Other ships, as you can see down here below, this is the Scripps Marfac with all of the ships there in dock. Those, some of the smaller ships would be for shorter voyages or voyages that didn't go too far out. So what are the sampling devices that we use? Obviously, we got to send something down to the bottom to collect materials. And for the seafloor collection, we use a lot of different types. And here's a few of them. Some of them are as simple as the salad tongs, which basically we send down to the bottom and it just grabs some sediment. And typically we're in these scenarios, we're grabbing gravel and rock that's difficult to sample by other means. In some cases, we're also dragging the bottom here in the picture there in the center left. That's a benthic sled, so that'll collect creatures and rocks, etc., that are on the surface as we drag it along. And then other cases, we use a coring device, which gives us a long tube of sediment. So we basically use a huge weight and drive it into the sediment, and then extract the plastic tube that's inside a metal tube, and that has this history basically of that ocean area, all recorded in the sediments that are in that tube. One of the instruments that I use quite a bit is called a multi-core, and its picture is down there in the lower left. And what we do is lower that TP down to the bottom, and there are 8 tubes that are just slowly pushed into the seafloor, and then we bring it back. We get about 2 feet of sediment there, and what we're really interested in is those top several centimeters of sediment that are relatively undisturbed. But when we need to take precise samples, that is when we need to really see what we're doing and actually take samples or pick out certain rocks, we can't easily do that from using those devices that I just showed you from the ship. We're sending those down in a cable that might be thousands of meters long, and there's currents in there. And so it's very difficult to pinpoint exactly where you're going to be able to land the instrument, the device to collect the sample. So we really need to be able to see what we're doing, be able to be there and place coring devices or grab particular rocks. If we want to do that, then we need to use other devices. And one of those types of devices that we can use is a remotely operated vehicle. And there's several different types that we've used in the past. And these remotely operated vehicles are attached to the ship by a cable. And so that cable basically will enable people on the ship to be able to send commands to the ROV, and using the hydraulic arms, which you can see pretty well in the ROV Scorpio there on the upper left, we can use those hydraulic arms to pick up rocks or to take cores or to do other things, and then load those materials onto the baskets or whatever we have on the ROV and bring it back. And of course, we can also have cameras and lights, etc., that we can operate from the ship. And again, we've got a tether, essentially a long cable where the ROV is attached. Oops, yeah. The most famous of the ROVs is Jason. Now, this is the second version of Jason. I'm sure there's been updates since that time. The ROV Jason is the most widely used in the United States ROV, and it also has, of course, those hydraulic arms in the front. And again, with Jason, you need to kind of decide what kind of samples you're going to take. And so if you're going to take cores like those in the upper left, they're designed so that the hydraulic arm can pick up those little T handles and put the core into the sediment, and then bring the core back and into the quiver, and then we bring the whole thing back up. And as you can see, there's a lot of wires, there's a lot of technology that goes into Jason, and there's a lot of maintenance required, as you can imagine. A lot of engineering and a lot of technology went into the design and to these upgrades that have happened for Jason. The picture down in the lower left is the control panel, or the control van rather, that's on the ship that is directing Jason's activities on the seafloor. And it's really very much like what you might imagine at NASA. So you've got all of these monitors that are telling you different things. You have crew members that are there that are operating the the arms. And then of course you have scientists that are directing the operation. Let's take a sample here. Let's move on. Let's do a little survey over there. And oh, what's that over there? Let's head over in that direction and have a look. And so there's a lot of coordination, a lot of activity going on there. And there's a lot of information coming in from the cameras and other monitors on Jason and on the ship. And there's a lot of other jobs in the control van as well. This is a former undergraduate of mine who is taking data and recording data, taking pictures, etc., and giving them names so we can find them later. So there's lots of different jobs for the scientists on board. And we do this in shifts, obviously, 24 hours a day, so that we're continually operating, because as you can imagine, this is a very, very expensive type of operation. When you have the ship and you have this high-tech equipment on board. And then, of course, eventually you have to recover it, and we can recover it during the day or during at night. You can see the light there. So we've got the light on Jason, so we can see where it is as we recover it back onto the ship. Now, the old saying that there's no substitute for being there applies in oceanography as well. So sometimes there are situations where you really need to send people down to the bottom, where they can actually look around and see with the naked eye what the seafloor looks like and what the samples that they want to collect might be. And so for these kinds of operations, then we use a submersible, a manned submersible. In this case, I'm showing you the Delta submersible, maybe the original little yellow submarine, if you will. And as you can see, it's a little bit of a tight fit. As you can see me crawling into it, these submersibles are not really designed for comfort, especially for the scientists. So in this particular case, the pilot is sitting up on a seat and looking through that little bubble at the top and driving the submersible. The scientist is laying on the floor, basically curled around his feet, and looking out the portholes that are near the floor. Now, this particular dive, I remember it well. It's my most unsuccessful dive that I've ever had. We were trying out a new piece of equipment to try to take some cores on the seafloor, and it just didn't work. And it was so frustrating. But this is my most memorable dive as well, because when we came up after this very frustrating experience on the seafloor, we came up in the midst of a pod of dolphins. And so I'm laying there and looking out, and these dolphins are looking at me, and they're swimming around and they're playing with the submersible, and I just didn't really want it to end. It was just magical and an amazing experience that I won't ever forget. The most famous, though, of all of the submersibles is Alvin. Alvin is a 3-person submersible, and the version that I'm going to be talking about was able to go to 4,500 meters. There's a new version now that's been totally upgraded, and it can go to 6,500 meters. And this month is being put under trials, and they're testing it out. So there's a brand new sub, and it was designed to where scientists could lay down, like in the previous yellow sub there, the Delta sub, rather than sitting, which is what we had to do for this one. So it's going to be a little bit more comfortable. One of my former students is going to be going out on one of those trial runs, so I'll be anxious to hear what she has to say about it, because she went down in one of these on one of my research cruises, so she'll be able to compare the two. And Alvin is most famous because of its role in the discovery of hydrothermal vents, so places where hot fluids are flowing up out of the seafloor, which you've probably seen on TV. But it's even more famous because it was instrumental in the exploration of the Titanic. So it's pretty amazing to be elbow to elbow in the galley and having a lunch and hearing stories from these people who were there when history was made at the discovery of the Titanic and the discovery of hydrothermal vents. It's pretty amazing for both researchers as well as students. So the alvin looks fairly good size, and it is, but a lot of that, when you take the fiberglass off, there's a lot of space in there. There's a lot of tubes and wires and flotation, etc. But it's the main part of alvin is this titanium sphere that's located in the front portion of it, as you can see in the cartoon down in the lower left. And again, this is a 3-person submersible. And so there's 2 scientists sitting on cushions, essentially. And the pilot is sitting on a chair looking forward. And that's the person that operates the arms and also operates the entire operation under the direction of the scientists. Now, what does it look like inside? Well, down in the lower right, this is looking forward. So that's the front porthole that the pilot would be looking through. And the seat there is where the pink pig is sitting at the moment. But the entire inside looks very much like this, where there's a lot of switches and buttons and there's wires going everywhere. And so it's very basic inside, and it's not very big. You can kind of think of it like sitting in a Volkswagen without the seats, essentially. Again, there's only room for 3 people. Now, one of the aspects of this is that there's only one place where only one person can stand up at a time. So there's enough gear in there so that you're kind of sitting underneath things. And there's only really one place in the center there where the hatch is, where someone can stand up. And I know one of the questions in your mind at this stage, so I might as well answer it right away, is what do you do about the bathroom? Because there is no bathroom in alvin. And alvin goes down for about 8 hours. So what do you do? Well, you have a plastic container and you ask people to look away. And if you have a strong enough bladder, you might be able to last the 8 hours, but I can tell you most novices will do that, but they regret it severely when they get to the surface and you're bouncing around with a full bladder. And you can always know when people have made that choice, because when they get back to the ship, they don't want to talk to anybody. They don't want to do anything. They just head right directly to the head. And we get to find out the stories and what they saw down there after they have emptied their bladder. Okay, so when you're going to dive in alvin, you kind of decide again, just like with the ROVs, what kind of equipment, what kind of sampling are you going to do? Are you going to take cores, such as what we've geared for there down in the lower left-hand side? Are you going to have bio boxes, those white boxes there, which have lids where you can put creatures inside, or samples that you don't want to wash away when you come back up to the surface? Now, what kind of sampling are you going to do? So this is the front of alvin, as you can see. So you can see that porthole that we were looking at from the other direction. In the previous slide, you can see cameras there. You can see the hydraulic arms, lots of lights, of course, because it's dark down there, and a sample basket there in the front that is somewhat limited. So you can't take everything on every dive. You got to make some choices. In some cases, you want to make choices about special equipment. So in this case, we were going down with some specific sampling in mind, and the special bush collector there that we have is designed to collect these bushes in quotes, of these tube worms that we're going to talk about a little later on. So this device was designed specifically to collect them. There's also slurp guns. These are basically like little vacuum cleaners, essentially, that can slurp up some of the surface sediments, some of the bacterial mats, et cetera, that we might encounter down there. And then we have BioBox. We also have special sensors. So in addition to temperature and salinity, which they typically have, we added oxygen to the sensor array so we could know what the oxygen, dissolved oxygen levels were. And we also had some bottles where we could collect water samples on this particular dive. So what's it like to dive down to the bottom of the ocean? And what do you have to do ahead of time? Well, you have to make sure that you demonstrate that you're not claustrophobic, because that would be a problem. It's pretty cramped down there, and you don't want to have a claustrophobic moment when you're at 4,000 meters below the surface of the ocean. And so also they tell you about a lot of different things. So you go through safety training. So if the pilot passes out, then you have some idea what to do before your adrenaline levels get too high. And they also tell you can't wear any jewelry, so no watches, no belt buckles, because when you're crawling into the small hatchway, they don't want anything that could potentially scratch that hatchway, because that might compromise the hatchway and allow water in, which would be a real problem at 4,000 meters. And so when you go aboard, you're hoping that the other two people that are going with you didn't have beans for breakfast, because the air gets circulated around and around, and that would be a fairly smelly dive if that were the case. They do have a CO2 scrubber, which basically helps to remove the CO2 in the air, so to keep it relatively fresh, you don't get a headache. And there's big canisters behind the chief scientist's head. Essentially, the way that this was set up, there's a lot of equipment by the chief scientist and not so much with the assistant scientist. So actually the assistant scientist has a little bit more of a comfortable seat, which you can see there. This is one of the students from Costa Rica that accompanied me on one dive, and you can see where her porthole is, and that she's sitting against a cushion there. On the chief scientist side, the porthole is actually down by your knee. And so you kind of got to be a little more, do some contortions in order to really look out. But it's certainly worth it to look out the porthole and see what you see on the seafloor. Okay, so when you're on the bottom, again, you might be choosing what to sample and what sampling gear to use. So the Niskin bottles, as I mentioned, are ones that can allow you to sample the water. In the bio box, you can put in anything that'll fit in there and close it off and secure it. And it'll also keep it cold, because those boxes are insulated, made out of insulated material that doesn't compress, so it's not styrofoam, and also keep that water cold surrounding the organism, bringing it up through many thousands of meters of water that's much warmer than the seafloor. And then there's scoop bags where you can basically just scoop up creatures and some of the surface sediments. And if you're collecting rocks and you're not worried about things washing, then open containers work really well. And then, of course, there's the manipulator, so you can pick rocks and pick creatures, and the operators are really, really good at this. It's almost like you have a human hand out there, the way they're able to manipulate those hydraulic arms. And so taking cores, we can precisely place those cores and then in the sediment and get exactly what we want and where we want it, and then put it back into these quivers that hold the sediment in as we head to the surface. So this just kind of gives you an example. We were actually doing some experiments here where we had to squeeze that tube, and you can see the arm being able to do that. But you can also see that we placed those tubes just outside of the clam beds, and then we put them inside the clam beds. So this we couldn't do from the ship using just sending devices down. We'd need an ROV, or we'd need a manned submersible. Now you might say, wait a minute, you're sending people down there and you're sending these ROVs. Why don't we have complete maps of the seafloor? Well, you have to remember, first of all, that it's really dark, so you can only see as far as the lights will extend. But then also, I've showed you this picture before. So up in the upper left, this is a picture off the coast of the peninsula of Alaska, where we were studying. So we mapped this area. And just for reference, Dutch Harbor, which you may be familiar with if you're watching that TV show about crab fishermen sailing out of Dutch Harbor. That's where we sailed out of. It's an amazing place. Bald eagles everywhere, as common as seagulls. And a lot of other, there were World War II bunkers there. It's just an amazing place. And out in the middle of the nowhere, essentially. And so we mapped this area, and we were using Alvin. And so looking at the canyons here that I'm showing, and these are huge canyons, and one of many, many, as I mentioned before, you can see the black line there with the dots. That's our dive track. And the width of that line is about all that we could see. And you can also see in that same diagram in the upper right, camera tow. So there was an area where we actually towed a camera vehicle, so it was taking pictures of the seafloor so we could see what it looked like in those areas. But other than that, that's all we saw. And that's all we've mapped in that kind of scale. And I can guarantee you that no one's seen that area before, and no one has seen it since, and probably not in any other area of those canyons. And so these are virtually still unexplored and unmapped, really. There could it easily be many, many features that we didn't encounter. And there's another example down in the lower left of another dive track where we kind of went along one of those contours. But again, it's just a small area that we can map and look at, actually see the way you would walk around up here on the terrestrial realm. So what can you see on the seafloor? Well, what kind of creatures are there? And what kind of habitats are there? Well, most of the seafloor is soft sediment. And most of the seafloor gets their food from the surface. And we actually call that marine snow. And so it's dead creatures and poop from little creatures all settling down. And in the lights of Alvin at the bottom, it really does look like a snowstorm. And if it's really productive, there's a lot of material coming down. And so hence the name marine snow. But that's where most of the deep sea gets its food from, is from the surface, material just floating, settling down to the seafloor. And in productive areas, there can be a lot of creatures that make use of that food. Here you can see a bunch of starfish on the seafloor. It almost looks like a starry night, only we're at about 4,000 meters off the coast of Alaska. Here you can see a sea urchin as it's making a way across the soft bottom. You can also see tracks and trails and fecal material of other creatures that have made their way across this soft sediment. Here's a multi-armed starfish. And this is a holothurian, a sea cucumber. These can be quite abundant in the deep sea, and I've shown another example of one up there in the upper right inset. These creatures are kind of interesting because they also have representatives in the shallow water, actually in tide pools and places, and they have a very interesting way of defending themselves. They not only empty their bowels, but part of their intestines as well. And this smelly delicacy is thought then, hopefully by the sea cucumber, to be a delicacy that a predator can't pass up. And so they'll start eating that and allow basically the rest of the holothurian to escape. But if you're someone that's, if you're a human, and you're messing with one of these from a tide pool, it can be a really smelly experience if you disturb these creatures a little too much. Well, so there's other creatures that you might not have thought of that can be really abundant on the seafloor, and those are single-celled creatures, and even single-cell creatures that you can see with the naked eye. So these are Foraminifera, and they make up a large portion of the biomass of the deep sea. Xenophyrophores, for example, are a type of Foraminifera that are huge. So these that you're seeing there are the size of your fist. So this is a single-celled creature that's the size of your fist, and they come in all shapes and varieties. I'm just showing one there in those insets below, but they can actually be quite abundant, and there's different types in different places. And so off in the Pacific and the Atlantic and North Carolina, and even in Santa Catalina Basin, just off the coast here, there are places that have a lot of these Foraminifera that you can actually see with the naked eye. They are not quite as big as the some of them are not as big as the xenophyrophores, but again, you can see these single-celled creatures at they're inches long in some cases. So there are some other creatures here, just kind of a smattering. So they're brittle stars off Alaska, so these are related to starfish. They're sponges, so these are a different type of sponge at 4,000 meters off Alaska. We encounter a number of octopi off Alaska and off Costa Rica in some of the environments that I'm going to show you a little bit later. And one of the most cool things, though, that I've seen, I've only seen it once in Monterey Bay, were these gastropod egg cases. So these gastropods were stacking their egg cases up in these little stacks that you can see there, and you can see the gastropods, the snails, all around there. There's a big anemone, and then there's a rockfish just making its way off the photo there. But those were pretty cool, I thought. Now, unfortunately, there's pollution everywhere, including in the deep sea, and especially even in the deepest spots, and especially in areas where there are shipping lanes. And so it's really disappointing, really frustrating, really maddening to see in these pristine, unexplored environments, to see trash on the seafloor. And it's not just the toxicity or the plastics degrading there, which are terrible things, but it's also just the presence of a hard substrate. Because remember, this is all soft sediment. So now you're introducing a hard substrates here, which allows other creatures that wouldn't normally be able to live there to come in and attach, etc., and to use as refuge, all of those things. And you're basically altering the ecosystem, altering the communities there by just the physical presence of trash like this chair. So as you've probably guessed, some creatures like soft substrates, but there are others that prefer hard substrates, and there are natural hard substrates. And when I'm going to be talking about some of those in more detail, but creatures like octopi, the octopus likes hard places as refuge, as places to hide, and also to ambush. Fish like those areas as well as good places to hunt. And of course, creatures that need hard substrates to attach to, like the anemone and those corals, those deep sea corals down in the lower right, those are off Alaska. Those require hard substrates. And so they're not going to be able to live in soft environments, soft sediment environments. Here's just a few more pictures, including a jellyfish, a jelly of some sort that's swimming around near the bottom, and that same holothurian also down in the lower center. And the rest of the creatures are on hard substrates, so corals, anemones, the octopus, some crabs, and some crinoids as well. So lots of different creatures making the hard substrates are their habitat, making communities in those environments. And those can be very different, as I mentioned, from the more typical soft sediment communities. Okay, so I want to talk a little bit about a special environment that we know as a methane seep or a cold seep. And these are places where methane is bubbling up out of the seafloor. And I'm not going to talk about hydrothermal vents. You've seen those on TV, places where there's hot fluids bubbling up. This is places where cold fluids are bubbling up. And in this case, I'm going to focus on places where methane is bubbling up. And the reason why the methane is there in the first place is it's buried organic material that is degrading and creating this methane gas that then makes its way up through the sediment and in some cases into the water column. But in some cases, as it's making its way up through, of course, it's so cold and there's enough pressure that sometimes the methane actually gets frozen into an ice lattice, basically, and we call those hydrates or clathrates. And there can be really extensive areas of clathrates or these hydrates. And there are off the coast, off the west coast of the United States and off the east coast. And of course, one of the major worries is that as the temperature of the ocean is warming up, that we will start dissociating those, and that then those that methane will be released. And of course, methane is a major greenhouse gas, and so that can be a major problem. In fact, in looking at the geologic record, it's been suggested at several places in the past where these methane hydrates have been liberated and greatly affected global climate. And it has a very distinct carbon isotope signal, and so we can actually see that that change, that shift in older rock records and older sediment records from the ocean. And so again, it's been suggested that these can play a major role in climate change and be affected by it, and then also affect it. So they're important places to have a look at. Well, what are cold seeps? Basically, these cold seeps, these cold methane seeps, are places where reduced compounds, and it's primarily methane and sulfides, are released into the sediments. And again, it's in the ambient temperatures, so they're at the temperatures of the cold sea floor. They're not hot. And what's interesting about these is that these areas are, the ecosystems that occur at these cold seeps are based on chemosynthesis. And what is chemosynthesis? It's basically the ability, the process of converting the chemistry of these fluids, methanes and sulfides primarily, into energy. So instead of photosynthesis, which is what we have in most of the terrestrial environments are based on, where plants are getting their energy from the sun and converting that into plant tissue. Of course, in the deep sea, there's no light, so there's no photosynthesis, there's no plants. But in these environments, there are specialized single-celled creatures, bacteria and archaea, that can convert that chemical energy into energy that other creatures can use. So creating carbon compounds that other creatures can use to build their tissues. And so they are the base of these autotrophic microbes. These bacteria, these archaea, are the basis of the food chain. They are the basis of the ecosystems in these seep habitats. Now I'm going to show you seeps that we discovered in several different places, including, you've seen this one before, including off Alaska, a couple of places off California, Eel River and Monterey Bay, and some places off Costa Rica, as well as some places off Oregon. And I want to talk about the creatures that occur in these seeps. And what sort of creatures live there? Well, there's mollusks like the mussels and the clams. And there's also these large worms that we're going to be talking about. and these bacteria. And in some cases, they come as bacterial mats covering the seafloor. Now, one thing I want to point out, because this picture really shows it well, up in the upper right, these clam beds, you kind of notice that they're kind of in lines. And the reason for that is that there are faults, there are fractures or faults in the rock below them that allows that methane to come up in those lines. And so those clams need to be where that methane is coming up. And I'll talk about why that is in just a little bit. But we've actually been able to map faults on the seafloor by using the distribution of these clams. So first, let's talk about bacterial mats. And one of my favorite methane seeps is in Monterey, these seep rings. They kind of remind me of the fairy rings, the tree rings that you see in the forest sometimes, if you know what I'm talking about. So in the center here, we've got a mass of bacteria, these bacterial mats, and they may be white or they may be colored yellow with sulfur. And then surrounding those are these clams that can't necessarily handle the toxicity and the fluid flow in the center, but really thrive in that outer part of that ring. Now, bacteria are not just in the mats. So they can form mats like you can see there, and like I showed you before earlier, but these microbes are also in the sediment, they're inside the rock, and they can be inside the tissues of larger creatures as symbiotic creatures, as creatures that are living together with these larger creatures. And in this case, there's mutual benefit between these microbes and the creatures whose tissues they're living in. And in any case, these bacteria make life in these seep habitats possible. So they're converting that energy, and they can do it in the mats, or they might be doing it in the tissues of some of these creatures that we're going to talk about. Another thing that these creatures do, so these bacteria, the chemistry that they're creating promotes the precipitation of limestone, believe it or not. So calcium carbonate is promoted and precipitated in these environments where these bacteria are doing their thing. And as a result, you can get huge accumulations of carbonate, of limestone on the deep sea floor, and in some cases even hills of carbonate that have accumulated over many years, and large chunks, the size of a small building or a bus. And these hard substrates, as we talked about, are very different from what most of the rest of the habitat is like. So it's not soft sediment, it's hard. And so different creatures are able to live there. There's a greater diversity. These are places that should be regarded as oases, essentially, of activity and of a source of food for both the creatures that need those bacteria, but also other creatures like fish coming in and feeding on some of these creatures as well. And so you can kind of regard these bacteria as like beavers. On the surface, we have beavers that convert a river into a lake, which then lake trout can live in, and ducks, etc. And the same is true here with these bacteria. These are keystone species that are able to affect their entire environment and affect the types of creatures that can live there. And this is just by their the presence and the promotion of the precipitation of these limestones. Okay, onto the clams. So the clams are the clams that we're looking at here, and there's a couple of different types. Have those bacteria within their tissues, and so the clam sticks its foot down and gets the carbon and also then uses its gills to bring in oxygen for these bacteria and that are in their tissues, and in return, the bacteria convert that into nutrients, not only for the bacteria, but also nutrients for the clams. And it works out very well for them. But of course, they can only live in these environments then where these reducing compounds like the methane seeps are occurring. So they're only in certain places where the methane is bubbling up. So these, the pictures, all of the pictures on the left are from Alaska, and the top there with the crab in the middle, that's from Alaska. And the one down in the lower right, that's from Monterey Bay. Okay, so the next creature I want to talk about are these tube worms. So these worms also have symbiotic bacteria in their tissues. And so the worm uses its gills basically to bring in oxygen, and then it sticks what its root-like features into the sediment and gets the other carbon compounds that from methane, for example, and sulfide that the bacteria can use, and then the bacteria in return provide nutrients. Now these worms can get up to 10 feet long. So this is a good way to do it. And that's their only source of nutrition, because these worms have no mouth and no anus. Now, it might be kind of nice not to have to go to the bathroom, but not to be able to taste your food, yeah, I'm not sure if that's a good trade-off, but it works really well for these worms. Now, these worms make a chitinous long tube that you can see there in the hydraulic arm, in this case of Alvin, and you can see the bush there in the background. So these bushes actually can hold over 1,000 of these specimens. We found one bush that was the size of a Volkswagen Beetle, and had an estimated, I think it was like 1,400 of these. And so these creatures, again, need to be in places where there's methane bubbling up, or where they can get access to that through their through one end, and then get oxygen in from the other. And they use hemoglobin, just like we do, to transport oxygen from the seawater into the bacteria and into their tissues. So their blood is as red as ours. And you can see some examples here where you can see a few hollow tubes, but you can also see them living in the tubes there. And you can see one where we've extracted it from the tube. And you can see one there that one of the tubes that we're holding in the lab. That's about 6 feet long or so. So these things are amazing creatures. And they're entirely dependent upon the bacteria in their tissues and the ability to then get those toxic compounds to the bacteria in their tissues, and then get the nutrients from those symbiotic creatures. One of the really cool creatures that we encountered off the coast of Costa Rica were Yeti crabs. And Yeti crabs were first found in 2005, so not very long ago. And in fact, our expedition really increased our knowledge about these creatures. There were only a few specimens known before, and one location known prior to our expedition. And we found large colonies of these creatures. So it was really fortunate that we were able to find these off the coast of Costa Rica. And you can see down in the lower left that these creatures have these hair-like projections that are coming off their carapace. And in life, these are covered with white bacteria. And you can see a close-up of that filamentous bacteria there in the upper right hand photo. And so this gives those creatures a very white look, hence the name Yeti crab. Their juveniles are down that look like the one down there in the lower right. So they don't have those hairs on their carapace until they get a bit older. But what was really cool when you're down in Alvin and you're looking around and all of the cracks and crannies of this limestone, you see these crabs in there, and they're all doing the Yeti dance, back and forth. And the Yeti dance, basically, they weren't all in sync, so not all of them were dancing to the same tune, I guess. But they're all dancing, and they're all swaying back and forth. And we think that the reason that they're doing that is to aerate the bacteria on their bodies. And we think also that they're benefiting from those bacteria, whether they're eating them directly or having some other benefit. We're not completely sure about that. But the speculation is that the dance is to keep those bacteria happy. Now, there are lots of other crabs as well and other creatures that I don't have time to show you, but they're not all, as I mentioned, necessarily endemic or associated with those methane seeps. These are oases again, so you've got other creatures coming in using, like the octopus, for example, that's using this area for a refuge from predators, but also using it as a place to capture food. So there's lots of creatures like that that are benefiting from these communities that are not necessarily have symbiotic bacteria associated with their tissues. And that includes these some of the other crabs. So this particular crab is not a crab that that is associated with a methane seep, but it's there and taking advantage of the food availability that's in these oases of methane seeps. So all good things have to come to an end. And so eventually your dive is done. And so your swimmer comes out and actually talks to you on a telephone that is located in the top of Alvin and basically guides you in, and you get back on the ship and you tell your tales. Then obviously also you have to sail home, or you get to land and fly back home. This is off Costa Rica, for example. So we met the ship, went out in a Zodiac and met the ship, and then went with a smaller ship back to Costa Rica, did a little bit of zip lining, and then headed home. So everything has to come to an end, as I mentioned, but you're going to be taking samples back, and we're going to be shipping samples back. And then you work on them in the lab, obviously. Okay, so at this point, I'd like to tell you a little bit about our marine program, our marine science program at CSUB. This is for students and teachers. Right up to this point, we've taken over 200 students and teachers out on research cruises off the coast of San Diego. And this is in collaboration with the University of San Diego and CSUB. So students from both places have gone out and interacted and gone out on these cruises where we actually collect real samples. We're collecting real research samples on real research vessels, real Scripps Institution of Oceanography research vessels. We've got a couple more cruises coming up, one in the spring and one in the summer. And I'm looking for participants. So if you fall in those categories, this might be a good opportunity for you to get some hands-on experience, because we treat all of the students on board like real scientists. They're involved in every aspect, from the packing to the setting up of the labs, to the deployment and recovery of equipment, to the processing and preserving of equipment and labeling and entering data, and then also doing research on these samples when they get back to the lab. And I might mention that there's some really good PowerPoint presentations that have been made by previous students to document their experiences. So what is it like to eat on board? What do the rooms look like? What is it like to work on a research vessel elbow to elbow with scientists and the crew? And so I would encourage you to go to our website, which is on the Scripps website there down at the bottom. Don't worry if you can't write it down right away before I switch to the next slide, because this is going to be recorded, so you can always come back to it. And if you have any questions or might want to participate in our marine program, hopefully even those that are not college age yet are thinking, if you're thinking about being a marine scientist, an oceanographer, then hopefully CSUB will be in your target. We're doing marine science here, and we're not just working off the California coast, or at least that's not the plan. We're going to be working all over the world. Okay, with that, I'll end the presentation and ask for any questions.
Can you see the ones in the chat box?
No, you'll have to read those to me if you would.
Okay, I can do that. Hang on, let me get the slider up here. First question, are some sampling strategies better than others to minimize disturbance to the environment?
Yes, absolutely. Our sampling is minimal. So the disturbance is, even to these environments, the disturbance is minimal, but there is deep sea mining that is really threatening a lot of habitats, including methane seeps and hydrothermal vents. They're minerals, valuable economically viable materials are precipitated in those environments, and manganese nodules, for example, and those are being destroyed for the purposes of mining. And since it's at the bottom of the ocean, people aren't really aware of that. Our sampling doesn't really affect anything because it's such small scale. But we do have to be very careful about, especially with the creatures, because some of those creatures are like some of those corals, for example, are 100 years old. And so if we're going to sample one of those, it's got to be a really, really good reason for us to sample a deep sea coral, for example. There's got to be some really strong scientific reason to sample that, because it's and some of those crabs are also quite old. So we're really cognizant of that, and we're really careful about those kinds of things. But I'll give you one story. In one of these seeps, where was it? It was off California. Might have been Eel River. Yeah, I think it was Eel River off the coast there on one of these seeps. On one of our dives, there were these sable fish. They're pretty good sized fish, 3-4 feet in length. And there was a whole school of them, and they were really curious what we were doing. And they were circling above us, and they were getting in the way, actually. When we were trying to take a sample, they'd come down and see what was going on. And we actually had to brush some of them away. That was one dive. The next dive, the next year, they were all gone, and the whole bottom there had been scraped with a net. So all of those creatures were gone. And again, these areas where there's hard parts, or where there's hard substrate, is an oasis of food, and so fish are attracted to those environments. So you do have to be really careful, and there is real concern about the conservation of habitats on the seafloor. For some of these, the hydrothermal vents, as well as these cold seeps, if they were on land, we would be treating them like Yellowstone. But since they're not, we're not doing that. Good question.
Thank you, Alexander, for that question. The next question is from Isolda. He asks, how long does it take to reach the bottom of the ocean, and how much time can you spend there for sampling?
Great question. Of course, it depends on the depth. So as I recall, when we were going down to about 4,000 meters off the coast of Alaska, it took about 3 hours down and 3 hours back. So since we only have 8 hours, we only had 2 hours on the seafloor. Luckily, they have a CD player, so you can listen to music, because once you get out of that fireworks zone, where you see all of those as you're going down in Alvin, once you start getting into the twilight zone, you start going through an area where you're disturbing these creatures that bioluminesce. And so you can see little strings and stars, and they're sparkling everywhere, and it's like fireworks. It's amazing. And of course, it's natural fireworks, but it only lasts a certain distance. And then you get out of that zone, and then you're in dark environments. And the Alvin doesn't put its lights on because it wants to conserve the batteries until it gets down to the seafloor. So you've got essentially 2.5 hours or more. And luckily, there's a bit of music that you can listen to, though the pilots can be pretty specific about what music they will allow in some cases. So again, it depends on the depth, but it can be up to 3 hours or more.
Thank you. Next question is from, scroll back up. There we go. From Kamala, have you ever seen weird creatures in the deep ocean?
Well, I mean, some of the creatures that I showed you are pretty weird. Yes, I've seen a lot of them, many that I didn't necessarily know what they were. Those deep sea fish that you see on TV, the big teeth and that have the lights. I never saw one while I was in Alvin, but we did bring one up in one of the instruments. It got caught in the net of the multicore in one case. So we did find those. Those aren't very big. They're only a few inches long typically, but they do have really big teeth. And if you take a picture up close, they look really menacing.
Lucas asks, what got you interested in marine science?
I got interested in marine science because I was interested in fossils and became interested in microfossils that live or that can be recovered and whose modern representatives live in the deep ocean. And so that means that I need to be a marine scientist in order to study them. And I find it more intriguing to study these places and creatures that haven't been really studied before, as opposed to the shallow water where you can go in the tide pool and everybody sees those. These deep sea environments are really important in a lot of respects, but in terms of looking at the fossils, the deep sea gives us these longer cores that can give us the history of how the oceans change through time and how climate has changed through time. Because again, remember, 70% of the planet is covered by seawater, and it affects and is affected by climate very, very dramatically. So we can get a handle based on the microfossils of how the environment has changed through time. And the deep sea is where most of that information is coming from these cores. And so I like to look, I like the interdisciplinary aspects of it, where you have to know a little bit about biology, a bit about ecology, about paleontology, about geology, about biogeochemistry, about physical oceanography, the currents, etc. You kind of have to know a little bit about all of that in order to really put it all together to synthesize it. And I really like that puzzle work. I like that detective work. And the fact that, when I'm out at sea, I'm elbow to elbow with some amazing people, like those people that were there at the discovery of the Titanic, and like researchers that I work with that are just brilliant people. And it's just wonderful to be in that kind of environment and have common goals while we're out there and discover new places. So a lot of those places that I showed you have never been seen before we went there and have not been seen again. And that's hard to do anywhere else in the world. Even in Antarctica, you can't really know that somebody else hasn't seen what you saw. But in the deep ocean, you can be pretty sure that that's the case.
Alexandra asks, have you ever seen a whale fall?
Oh, absolutely. In fact, I've studied them. So I haven't, in Alvin, I haven't really seen them, but I did get some samples from, I've seen some bones on the seafloor, so, but they weren't complete whale falls. So I guess I should back off on that a little bit. But I have studied some of those samples. That was taken by a colleague who does study them. And they're fascinating places. And actually, because they're generating so much organic material, and because the bones are so generating so much oil, they actually create a reducing environment, very much like a cold seep. And you actually get some of these creatures that we find at cold seeps and even in hydrothermal vents living in and around whale falls.
And Lydia asks, what cool field trips would you recommend for a younger student interested in learning more about marine science?
Ah, great question. Okay, well, so I would recommend aquariums, for example. So like the Monterey Bay Aquarium, but just going to the ocean, going to places where you can look at the tide pools. There's some really rocky environments to the north of San Francisco, near Fort Bragg, for example, where you, at the Sonoma coast, where there's a lot of rocky environments where you can actually have a look at what's in the tide pools. So I would recommend those kinds of field trips. So aquariums and going to places where you can actually see tide pools and see what's in them, which typically is more rocky environments than the typical sand type beaches where most people go. I might also recommend some of the fossil sites. So the Sharktooth Hill, for example, there's marine fossils there. That's from a previous sea that was in this area that covered part of the Central Valley in the Miocene. So if you're interested in the history of oceanography and paleoceanography, then that might be a good place to go.
That's all the questions from the chat. I'm now going to allow participants to unmute themselves. We'll see if there's any more questions.
Okay.
Okay, if you have any questions, please unmute yourself and pose your question. I think we've covered it all then.
Great.
Thank you, Dr. Rathburn, for your time today and for sharing your knowledge with us. The museum would love to have you back with us again soon.
Well, thank you. I really appreciate it, and thank everyone for attending and participating. I enjoyed it.
So long.